Okay, this is the DNA unit. Here's some fun facts about DNA. Each cell has about 7 feet of DNA, and of course, chromosomes are arranged in pairs, as we've been talking about. So at first, in the early 1900s, scientists thought that proteins were genetic material. They had done some experiments, and they just really did not think that nucleic acids were diverse enough to account for all of the traits that humans have. So then later in the 1920s, scientist experiments determined that proteins are not genetic material. And then by the 1950s, experiments did find that DNA carries the genetic information, and then it took a little bit longer to find out the exact shape and the dimensions of DNA. So these first couple experiments I'm going to go over are really just to show you or to tell you how they came to discover that DNA was the genetic material. So at first, Griffith was experimenting two different types of bacteria that causes pneumonia, smooth versus rough. By the way, these are not on the notes. This is just for your information. So only the S form is deadly. So by injecting the strains into mice, Griffith discovered what he called the transforming principle. So this would be the rough strain, which is it's non-virulent, and then the smooth strain, which is obviously smooth strain dies. They did heat-killed smooth strain plus rough strain, and that mouse died. What they later found out was that bacteria can trade DNA. So basically, the living bacteria were taking the dangerous part of the dead bacteria's DNA, which is a whole nother interesting experiment as well. Then later, Avery came along and determined that DNA was that transforming principle that Griffith had discovered because his chemical test showed that no protein was present. So here's what he did. He took that mixture of the S bacteria, then he took enzymes, and these enzymes cut up. This one cuts up DNA, this one cuts up RNA, this one cuts up lipids, this one cuts up proteins, and we don't really know what other is. But only the mouse where the DNA was cut up lived when of course, that S strain bacteria was added because proteins were not the genetic material. And then the Hershey-Chase experiment went on to just look at the same thing, basically. They just went on to prove that if you look at this, they took sulfur-labeled protein, which is in red because viruses have protein on the outside, and they injected it into cells, and they said, nope, look, we didn't find it. And then they did the same thing with a phosphorus-labeled DNA, because DNA has phosphorus. Remember, only proteins have sulfur, which is why that worked. DNA has phosphorus, and they took it out. Or the viruses release the DNA into the cells, and then after centrifugation, they found that. So here's the main idea of DNA. Humans have 46 chromosomes, 23 pairs. You get one copy from mom, one copy from dad. Each chromosome is made up of genes, which are made up of DNA. Sections of DNA that code for proteins are called genes. So DNA is deoxyribonucleic acid. It's made up of monomers called nucleotides, and each nucleotide has 3 parts. It's got the phosphate group, the sugar, and the nitrogenous base. So one molecule of human DNA contains billions of nucleotides, but we've only got 4 types of nucleotides, and all that changes is the nitrogen base. So the phosphate and sugar is the same for everything. Nitrogen base is the only thing that changes, and those bases are A, C, G, and T. So at first, scientists thought that DNA was made up of equal parts of each nitrogenous base, but later experiments showed that the amount of A always equals T, and the amount of C always equals G. So Chargaff, which was one of the scientists who pioneered DNA studies, came up with the rules. He just basically said that A always pairs with T, and G always pairs with C. This picture and how we know how they fit together, they didn't know this until much later. But they were just going off the pieces of what they knew at the time, even though it may seem obvious for us today. So DNA nucleotides are joined together by covalent bonds on the sugar-phosphate backbone. So these are covalent bonds in between each and every sugar and phosphate. And then the DNA bases are held together with hydrogen bonds. So all of those dots in the center are hydrogen bonds, if you remember those from the water unit. So based on Chargaff's rules, given one side of the DNA, figure out the other side of the helix. If we have T, A, C, G, what will this other side of the helix be? Should be A, T, G, C. You can do the others on your own, and then we'll go over them in class. So the 3D shape of DNA came about through various experiments. Rosalind Franklin and Maurice Wilkins were studying DNA using X-ray crystallography, which is where they bombard DNA with X-rays, and the atoms in DNA diffract the X-rays in a pattern that's captured on film. And based on the structure of the atom, what they capture on film is unique, and it can tell scientists a lot of information about the structure of not just the atoms of a molecule, but the way those atoms are arranged. So this was their famous, the famous photograph, and this, everything basically in this X shape like this, that indicated that it was a double helix. And we can do a demonstration in class to show how they would have gotten a double helix from this shape. So Franklin did all of these, this research, and at the time in the early 1950s, it was not very common for women to be in labs, and they weren't necessarily happy that she was there. They thought she should be an assistant instead of a scientist, like that she actually was. So Maurice Wilkins took her data, he took all of her x-rays, and he gave them to Watson and Crick, and they did the clues that they needed to figure out the 3D structure. She did not know about this, and she was going about figuring out the mathematical calculations, which could have taken, I believe it was about a year to figure out. Meanwhile, Watson and Crick just hastily threw everything together, and they got all the credit for it. It wasn't until later that Rosalind Franklin, is somewhat given credit today. So basically in their lab, Watson and Crick made models to figure out the structure of DNA instead of doing those mathematical calculations that Franklin was doing. So they placed the sugar phosphate backbone on the outside and the bases on the inside. And then they found that if they paired double ring nucleotides, which are A and G, with single ring nucleotides, they fit like a puzzle. And this was their early structure. So the bases are in the center, and then like this would be a ribose, and so on. So DNA has to be replicated because your body produces billions of new cells every day. And each time your cell divides, DNA must be copied first. Your cell can do this in about 6 to 8 hours, which is pretty impressive considering the billions of nucleotides. So it's very important that DNA copies itself exactly, as errors can result in mutations which can cause disease. DNA replication is carried out by proteins. Like we've talked about in other units, proteins run the world, specifically enzymes. Enzymes are key in DNA replication. Replication ensures that every cell has a complete set of identical information, which is really important, especially in mitosis. So step 1 of the DNA replication, helicase, which is an enzyme, unzips the double helix, like unzipping a backpack. So basically it latches onto DNA and it breaks those hydrogen bonds, almost like you would unzip a jacket. And then the second step is, now that that's open, all these free-floating nucleotides, they have plenty of free-floating nucleotides, and they're going to pair one by one with bases on the template as they're exposed. It's almost like it's a magnet. They would be attracted to that, and they would just automatically pair, because DNA wants to pair together. It's just the way the molecule works. So it would just try to connect on almost automatically. And then in step 3, DNA polymerase, another enzyme, is just going to bond those nucleotides together to form strands of DNA. So it's going to help put those things together. And then our result is 2 identical molecules of DNA. So this DNA process is called semi-conservative replication because the old strands are conserved and they serve as complementary strands for the new strand. So there was another experiment to show semi-conservative replication, and basically what they did was take radioactively labeled DNA, because we can find out, or it was the backbone, we can find out a lot of stuff just by using radioactive labels, and then they put differently radioactive labeled DNA in, or nucleotides in, and they saw what happened, and basically their new daughter strands were a mixture of each type of radioactively labeled DNA. Or nucleotide. It was nitrogen that they had attached the label to. And this is what they got. So this is semi-conservative because half of each old strand is conserved, and then each new strand also has half of a new strand. So what happens when DNA is copied incorrectly? Mistakes happen. Any change in DNA is called a mutation. Mutations can be harmless or they can cause disease. Sometimes mutations can even be helpful to an organism. These are videos on DNA replication, which will be posted on your Canvas page. So we have one other type of nucleic acid, and it's called RNA, or ribonucleic acid. RNA is similar to DNA, but there's a few key differences. So RNA uses ribose for its sugar instead of deoxyribose, and it has the bases guanine, cytosine, adenine, and uracil. Remember, the DNA has adenine, guanine, cytosine, and thymine, so it's the uracil that's different instead of the thymine. RNA is also single-stranded. Here's a picture of RNA, so you can see the pentose is the same. It's still going to look the same. There's really just an oxygen difference, so ribose has an extra oxygen compared to the DNA, or deoxyribose, which does not have that oxygen. But as far as we're concerned, looking at the overall shape on the outside, they're going to look the same. And these are just the 4 bases, and it's just single-stranded. So it looks very, very similar to DNA, and it's slightly different. And RNA is going to have a bigger role in the processes that we talk about next, like protein synthesis.